Over to Stanley.
Okay, so I'm going to talk today about anosmia using some information from human studies and also from our mouse work. And if I have time, I'll go into some neurological sequelae that we've been observing in our infected mice. So to start off with explaining why I care about this. So actually, before I start, this is a figure that was given to me by a tattoo artist in 2020 who was put out of business, obviously, for obvious reasons by the pandemic. And he started doing prints instead of tattoos. And he thought I'd be interested in this particular image, which I think really summarizes what the pandemic has done to us. So getting onto the talk, the thing that I think is most important as we get over the acute phases of COVID-19, and of course, there's a question about how well we're doing that right now with all the new variants coming out. But long COVID is a syndrome that I think is going to occupy the healthcare system for many years. And it clearly affects many people who survived COVID-19. And it can occur after mild or severe disease, still being defined, mostly is affecting the brain and the lungs, but people have fatigue and other manifestations. So I think we really need to understand what's going on here, especially since virus is not generally detected in most human organs at autopsy, except the lungs. And even then, often not, because it's many weeks after the initial infection. And so I've been interested for many, many years in neurological sequelae of coronavirus infections. And in terms of what we see with COVID-19, during the acute phase, we see seizures and strokes and anosmia, the loss of sense of smell, ageusia, the loss of sense of taste. There's reduced consciousness, there's brain fog. And these resolve in most people, but in some people, they don't resolve. And so those are the people that we're most concerned with here. Olfactory dysfunction is one that's been found in many, many people and often resolves, not completely, as I'll discuss. And it's something that we think we can study using mice. Sometimes this evolves into parosmia, which is anosmia is the loss of sense of smell. Parosmia is when one smells something, sees something and smells something else. And phantasmia is when one has senses a smell that's not even apparent in the environment. So this is one of the first slides talking about anosmia. This is actually a report from Amazon. And so what it shows is that in 2020, in October, November, and this is in the United States, there was an increase in number of people who on reviews of scented candles said, hey, my candles don't work. This candle has no scent. And this was the time when COVID-19, one of the first times it appeared in the US at high levels. And so this was an indication that anosmia was present at a population level in the US. And this is another example. This is a woman in March 2020, right at the beginning of the pandemic, who one day woke up and couldn't smell or taste anything. She was perfectly fine otherwise. She had been exposed to her daughter who had been in California where there was COVID-19. And she had also been at a wedding, and this was before people in the Western countries used masking. So she lost her sense of smell, but recovered most of it at 14 days, but by several months later, still had not completely recovered. This for her was actually very good financially because she really liked fine wines and could no longer tell the difference between a fine wine and a not so fine wine. So, and I don't know if she's recovered completely now. Several months later, we were asked to determine whether she had possibly had COVID-19. So we did antibody testing and found that she had a low level of neutralizing antibody, 1 to 48, and we did T-cell assays and found that she actually had T-cell responses to the peptide pools from SARS-CoV-2. So we're pretty sure that she had been reinfected. So why do we care about anosmia? Well, there's suggestions, not proven, from the literature from other diseases that chronic anosmia may be either a sign of impending neurological dysfunction or neurodegenerative diseases. Certainly after, this is complicated, but after the pandemic of 1918, there was a disease called encephalitica lethargica, where people had some of the same kinds of signs that people now have who had COVID-19. Originally, this was considered to be part of the pandemic and a manifestation of a long-term effect. More recent studies raise questions about this, but still, this is the kind of thing that we worried about. Certainly, Parkinson's was associated with people who had the influenza, been infected with influenza in 1918. So this is why we care about this. And what it means is that people who have persistent anosmia need to be followed to make sure that if they're starting to develop signs of cognitive defects or other long-term sequelae, that they need to be treated appropriately. So the thing about SARS-CoV-2 is that what we knew from the beginning, both from human studies and animal studies, is that ACE2, the receptor for the virus, is not really expressed on neurons, but it's expressed on sustentacular cells, which is a cell that very few of us knew much about. These are supporting cells in the olfactory epithelium. They're believed to be involved in homeostasis and various other things, but it's not really so well defined. Let me get a pointer up here. So these are the cells here. So one of the key questions, which I'll keep coming back to, is if a cell is infected, sustentacular cell is infected, how come one has anosmia, which is very controlled by olfactory neurons, which are not infected? So to address these questions, while we can do some things in patients, clearly there's a lot of things we can only do in experimentally infected animals. We knew one of the things about this pandemic that's was apparent from the beginning is that many different kinds of animals could be infected by the virus: hamsters, macaques, ferrets, and cats. But more recently, the concern is the animals that weren't initially infected, like mink, deer, and hamsters, could be infected in the wild, as it were, and these may be reservoir for infection back to humans. We know from minks in the spring of 2020, we knew minks were infected and could then reinfect humans. The viruses that are transmitted like this never really took off, so they may have been better adapted to mink than to humans. In the US, we have a really incredible situation with deer, where in two studies, one in Ohio and one in Iowa, in Ohio, I think it was 15%, in Iowa, 35% of deer were positive for SARS-CoV-2 RNA. I don't really understand how that works, because the deer across Iowa don't all congregate anywhere. But it's certainly a concern that this could be a pool for future transmission to people. So we've always been a mouse lab, so what we wanted to do is study these kinds of phenomena in mice. And we had, because of our years of working on coronaviruses, we had many approaches to doing this. We had developed a system where we used adenovirus expressing human ACE2 to transduce mice for infection. This was at the time when ancestral strains were available that did not directly infect mice. There were human ACE2 transgenic mice, which we had developed as part of SARS, and then human ACE2 knock-in mice, which we and others developed. We're still characterizing the ones we developed. And there are some interesting aspects to this that we're very, very slowly finishing up. And then the other thing to note is that SARS-CoV-2, the ability to infect mouse, to use mouse ACE2 to enter cells, required only a 1 to 2 amino acid change in either mouse ACE2 or SARS-CoV-2. And I think most people know that many of the variants can directly infect mice. So in one of our first studies, we looked at the noses of mice, the K18 ACE2 mice, because that's the first ones we had, that we looked in the nose for infection of cells. And what we found is much like we saw in humans, which I'm not going to show the human data, is we found these cells here, these sustentacular cells were infected. You can see that in panels M, N, and O. In the nose, we saw lots of infection of the maxillary sinus. We saw respiratory epithelium infection as well, but I'm going to concentrate on the olfactory epithelium. And what we found in the mice, just like in humans, is that the cells were infected. The red cells here were the sustentacular cells. The green cells, which are neurons, were not infected. So just like in humans, only sustentacular cells were infected. Then we did experiments, and this was Abhishek Verma's work, asking, do these mice have anosmia? And there's two kinds of tests that we did. One was to ask mice to distinguish between novel and familiar scents. We did this with female mice, where the female mice preferentially are curious about the novel scent. Male mice, we do the same thing, but instead of using novel and familiar scents, we use male and female scents. And in both cases, particularly in females, we saw that while normal uninfected mice could spend much more time with the novel scent, after infection, they spent equal amounts of time with both scents, showing that they actually had lost a sense of smell. And then we did the same thing with cookies. This is one of our favorite childhood books. If you give a mouse a cookie, they often ask for milk, but these mice did not. Instead, these mice went probing for milk, not for milk, rather for cookies in the bedding. And when we did this, we saw that mice that were infected could not really find the cookies. These are female mice again. And when the ones that could took much longer to do so. So this was, again, another indication that the mice had anosmia. And so this was our first study. And then we went on to show that a virus antigen could be detected initially in these sustentacular cells, but by day 15, it was cleared. Same thing with RNA. One looks at the scale here. At days 3 and 6, the scale is much higher than what one sees even at day 12. And then by day 30, there's virtually no RNA left. So why do mice and people develop anosmia if olfactory neurons are not infected? And we don't really know the answer to this, but as I mentioned before, sustentacular cells control neuronal homeostasis. They may be immune-mediated damage. In other words, infected cells induces some sort of immune response, and that causes damage. The other thing that, which I may come back to, is that the other thing that sustentacular cells do is, so when mice or people smell something, an odorant binds to a receptor on a cell. And then to desensitize that cell so that one can then smell something new, the receptor odorant complex is taken up by the sustentacular cells. So it's possible that the sustentacular cells are not removing odorant, and that's the reason there's anosmia. And we have some hints that this may be the case, which I hope to get to. So one of the things that Abhishek looked at is the way olfactory receptors work. Each one has a cilia on it, and each cilia has one receptor. And what one can see here is in a mock-infected animal, there's a nice layer of cilia all around the olfactory epithelium. However, at 6 days after infection, it's really disrupted, even though the number of infected cells is not that great. And by 15 days, it's mostly recovered. And then this is one that I think, this is a curiosity that I think that I don't fully understand yet. But basically, this is a measure of olfactory receptor mRNA. So not the protein, but the mRNA. And what one can see here is in all cases at day 2 and 6, expression of the mRNA is greatly diminished. So receptors are not being made anymore by these cells, which could, of course, contribute to anosmia. By day 12, it's back to normal, even though the cilia mostly normal then, but not completely. And then we saw some inflammatory molecule expression. And I think there's some really interesting things here. Look at interferon beta. Instead of being high at day 2 and 6, it's actually very low. Same thing with interferon lambda shown here. And with ISG15, an interferon-induced gene. And on the other hand, a few NF-kappa B type genes, CXCL10 and IL-6 are both increased to some extent. So the inflammatory pattern may be consistent with what we often see in SARS-CoV-2, less of an interferon response and more of an NF-kappa B type response. And the one thing that we did see that was consistent is this change in myeloid distribution. So I'm not showing it, but we saw no change in numbers of inflammatory cells in the olfactory epithelium when we looked at myeloid cells and T cells. But if instead one looks at the cells which are normally present in the olfactory epithelium, and remember they're here because the olfactory epithelium is exposed to the environment. So these cells are sitting in the basal layer, but what one can see is after infection, even though the numbers don't increase, they tend to move up to the front of the, or to the surface area of the epithelium to be near to the infected sustentacular cells. So this is one contribution that may contribute to the anosmia, though the direct connection is not something we know very well yet. So the second approach, which, and there's a reason I'll describe this, is to try to study this instead of using K18 mice, which have the problem always of potential brain infection. Sometimes there's a brain infection, sometimes not. It's much better than with SARS-1, where we always saw a brain infection. But SARS-2, there's not so much brain infection all the time, but still, it'd be nice to have no brain infection. So what we did, based on work of others, we developed a mouse-adapted virus, which would be useful for studies of anosmia. And very early on, in early 2020, two groups, Ralph Barrick's group engineered 2 mutations into the virus that allowed it to infect mice. These are ancestral strains again. And Gu et al. Found a single mutation, N501Y, which has become very important in other settings. But he found this in populations of the virus as early as January 2020. This mutation didn't become important until it was then selected in context with other mutations within the S protein. But we know that this mutation is important in the beta strain, beta strain, the alpha strain, the P1 strain. It's also present in Omicron. So and it's believed to be very important for virulence and transmission both in people and in allowing mouse infection. So we developed a virus using reverse genetics with the 501Y mutation. We passaged it 30 times in BALP-C mice, and we ended up with a virus that is quite virulent. And we've already published some of this work. This work was done by Roy Wang and Jian Zhang. And this virus, in theory, should have been available in BEI about 7 months ago. We're still working on getting it there, and we hope it'll be ready by late September. But it's very useful because it really causes quite a virulent infection without a brain infection. So when one looks at olfactory receptor expression, now what we found is that these mice also developed anosmia. I don't think I'm going to show that, but what's really interesting is the pattern of olfactory receptor expression is different than what we saw with the K18 mice. So in some cases, we saw a decrease in the amount of olfactory receptor expression, but interestingly, at later times, instead of going back to the same level, there was hyperexpression of these receptors. And you can see that across the board. Some are even more obvious than others. Here's olfactory receptor 45, which I don't know why it wasn't detected in uninfected animals, but it's very high in several of the infected animals. And the reason I think that this is interesting is that this may be an explanation of how people get parosmia, how they smell something else, they look at something and it smells like something else. So you look at a piece of chocolate, and maybe you should smell the chocolate, but instead you smell, a cigarette, because it's crossing of the information. So this may contribute to the parosmia that some people have. And the other thing is that in these same mice, we then did some of these similar analyses that we did in the K18 mice. So we looked for cilia expression. One can see here that there's, again, great loss of cilia, of a protein associated with cilia, and it's back by day 15. On the right-hand side, if one looks for cilia morphology, one can see in mock infected animals, the cilia are normal in appearance, but after SARS-CoV-2 infection, they actually look quite ragged. And similarly, we look for expression of molecules important in signal transduction. And these were also diminished in mice that had been infected with SARS-CoV-2. So, to very briefly summarize where we're at. So we think there's infection of sustentacular cells, maybe inflammatory cytokines matter, maybe there's poor uptake of olfactory binding protein with the odorant. This results from sustentacular cell dysfunction. Now, what happens after a signal leaves the olfactory epithelium? Well, it goes to the olfactory bulb, and then it's transmitted to the rest of the brain. And one of the things that happens in the olfactory bulb is these signals cause the expression of a molecule, tyrosine hydroxylase, which is important for, among other things, dopamine expression. So what Abhishek did next is look for tyrosine hydroxylase expression, because this should depend on down signals from the olfactory epithelium. And what one can see is that there's more staining here in the mock-infected animals. And when he quantified it, there's a statistical decrease in the number of cells expressing tyrosine hydroxylase. At the same time, there were minimal differences in the olfactory bulb in inflammatory molecule expression. And there's a little bit of viral RNA. This is about the only time in these mouse-adapted infected mice that we saw any virus anywhere in the central nervous system. We never detected virus in the brain. And this is all viral RNA, never infectious virus. And then more interestingly, we did the same experiments at day 120. And what one can see here, this is a little hard to tell from this figure, this immunohistochemistry, but when Abhishek quantified it, cells, he found there were fewer cells expressing tyrosine hydroxylase even as late as 120 days, and that the amount of RNA was also greatly decreased. At the same time, curiously, there wasn't too much change in many of these inflammatory mediators, but now there was an increase in interferon beta in the bulb, even though there's no virus there, and it's not really clear to us why the interferon beta would be increased, but we did see some. These other molecules may be increased, they weren't statistically significant. So some give hints of, in some cases, of increases. So in summary of this part of these experiments, we think these mice are useful for studying anosmia. female mice develop more severe anosmia than males, even though COVID-19 acutely is more severe in men. Anosmia and other long-term COVID effects appear to be more severe in females, so our mice may be mimicking that. I've gone over some of the mechanisms, so I won't discuss that anymore. And so what happens, what's going on here? Well, the olfactory system is not simply just smells. Certainly that's extremely important. There's the frontal cortex is involved in conscious perception of smell. The hypothalamus and amygdala deal with the emotional and motivational aspects of smell, and that, of course, would be lost too if your sense of smell is gone. And finally, the hippocampus is involved in odor memory. So there's many effects across the brain if one cannot smell items properly. And the other thing that's really curious is how so there's been postulation that in the brain of people who have long COVID or have brain fog, that even though we don't detect virus in distal parts of the brain, could there be a movement of inflammatory signals or even virus products that are below the level of detection from olfactory epithelium? This is something that we're thinking about, and other people are as well. There's some experimental systems that suggest that inflammatory signals can move across the brain. And the olfactory system is connected to many, many sites. In addition to the 3 that I have listed here, they're connected to many distant sites in the brain. So one could imagine if inflammatory signals could be transmitted across the brain, it would have profound effects on many aspects of neurological function. So just to finish on the last 3 minutes that I have here, one of the things we've been interested in is the substantia nigra, because this is important in Parkinson's disease, and based on the flu work where there might be a connection between flu and Parkinson's, we wanted to see if there were effects in our mice in the substantia nigra. So these neurons are important for controlling body movement, and they're dopamine rich, and we of course already have looked at tyrosine hydroxylase. So what Abhishek did next is he looked for tyrosine hydroxylase expression in the substantia nigra, and what he found was that in the number of cells was not statistically different, but was decreased at these later time points. And tyrosine hydroxylase, this was a complicated experiment, even though it looks simple. This is measuring tyrosine hydroxylase RNA. And because one's doing it in the BSL-3 and one has to cut out the substantia nigra, this may not be done precisely. So there's no difference here, but I think it's partly technical. I bet there would be if we could better define the substantia nigra before we isolated it from a mouse brain. When one looks for proteins such as dopamine transporter that could be affected, one can see great decreases at 120 days in these SARS-CoV-2 infected mice. And then this is a human correlate doing the same thing in humans. And looking at this substantia nigra, we've obtained autopsy samples that listed, this lists 20, but there's actually 22. And when we compare the 22 samples, there's lower levels of tyrosine hydroxylase positive cells in the COVID-19 autopsy samples than we saw in controls. So this is a hint that the p-value is not, it's only 0.11, but it's a hint that we're on the right track here. I should say if we take out all the patients who are under 55, which turns out to be 1 from each group, this becomes statistically significant. And then finally, the last thing that Abhishek did is he looked at a test for sociability, which is one of the things that's important in Parkinson's disease. And what one can see here is that where mice, here what we do is we ask whether this mouse would rather visit a strange mice or go to an empty cage, and usually they go and visit their friends over here. And what one can see is that the normal mice are more social, that's the red bars, but as with time, these mice are not terribly social, even up to 100 days post-infection. So we are doing many behavioral studies, and we're continuing with this. And so the last thing here is, what do we need to learn? We really need to learn what's going on. What's the effect? Is this transmission of inflammatory signals? Is the virus there for a period of time and then clear, but still having effects? Are there effects in endothelial cells and macrophage activation? So these are some of the things we'd like to address. This is a final figure. This was given to me one of my friends in Hong Kong, showing that even though 99.9% of COVID-19 has been terrible, at least it's resulted in a pretty picture. And with that, I'd like to stop and take any questions.